Good afternoon, students. In continuation with the chapter, that is chapter number 6, molecular basis of inheritance. In our previous video, we have talked about the transcriptional unit, and today we are going to talk about how transcription proceeds. What is the mechanism of transcription? But before advancing towards the mechanism of transcription, first let us now discuss what is a cistron and how is a gene defined. As we all know, that a gene is present on the DNA molecule, or we can even define a gene as a sequence of DNA, particular sequences of DNA which codes for particular polypeptides. It may also be defined as sequences of DNA coding for polypeptides, and accordingly these are also termed as cistrons. Clear? These cistrons, they can again be divided into monocistronic and polycistronic. Okay, when we are going to talk about the polycistrons, these occurs in bacteria and prokaryotes. But when we are talking about the monocistrons, these occur in the eukaryotes. And when we are talking about the monocistrons, these monocistrons, they have got split genes. Okay, these split genes, these are not the continuous gene sequences, but they are containing split genes, and these split genes are again categorized into two forms, that is the exons and the introns. When I am talking about the exons, these are those base sequences which are transcribed, which are transcribed in a mature RNA segment. Okay, and introns are those sequences which do not get expressed and which cannot be obtained in the mature RNA sequences. Those which do not get expressed and are not obtained in RNA sequences. Clear? So here we have talked about the gene sequences and what are the various types of the cistrons, that is the polycistrons and the monocistrons. Now we are going to proceed ahead with the actual mechanism of transcription. So when we are moving ahead with the actual mechanism of transcription, the tools that are required for transcription they are: number 1, DNA dependent RNA polymerase enzyme. Okay, then a DNA template strand, Clear? Then third is all the 4 type of nucleosides, all the 4 type of ribonucleosides. And what are these 4 types? Number 1, adenosine, triphosphates, Second, guanosine triphosphate. Clear? Third, cytidine triphosphate. And fourth is, in place of thymidine, it will be uridine. It is uridine triphosphate. So these are the four type of the ribonucleosides. Then the fourth, it is required the divalent ions like magnesium 2+ and manganese 2+. So these are the four tools which are required during the whole of the transcription process, and this is DNA. The number first is DNA dependent RNA polymerase enzyme. Second is DNA template strand on which the whole process of the transcription is going to proceed further on, and the third is all the four type of the ribonucleosides, and the fifth is the divalent ions like magnesium and manganese. Clear? And the process of transcription, it's divided into three processes. That is, first is initiation process, second is elongation process, and the third is termination process. Now how does this take place? We are going to talk about this in eukaryotes first. So when we are going to talk about the first one is initiation process. Initiation process. When we are talking about this process, as I hope that you all remember the transcriptional unit diagram that we have learned yesterday. This is the promoter gene, this is the terminator gene, this is the promoter, this is the terminator. Clear? This is structural genes. This is the coding strand. This is the antisense or the non-coding strand. Antisense or non-coding strand. Clear? So this is the diagram of the the transcriptional unit. Now what happens as soon as the initiation process starts, an enzyme which is known as the DNA dependent RNA polymerase enzyme, it comes and get attached to the promoter site along with some of the initiation factors. Now how does it come and attach itself to the promoter site? Because there are certain type of the factors which are known as the sigma factors, particularly in the prokaryotes. They are found particularly in the prokaryotes that can be very well easily identified by the DNA dependent RNA polymerase enzyme, to which it comes and binds up. But when we are talking about the eukaryotes, there are certain type of the transcriptional factors that are present on this particular type of the promoter site, which is present on the 5' end of the antisense codon. Clear? So the DNA dependent RNA polymerase enzyme, it first identifies those transcriptional factors and comes and binds up to this particular region of the promoter site. Clear? Now as soon as it comes and binds up, there are certain of the changes that takes place. First change that takes place is phosphorylation of the various ribonucleoside monophosphates. Clear? And this is achieved by, second phase is phosphorylation process. Phosphorylation process. Clear? During phosphorylation process, an enzyme which is known as phosphorylase is required, and it initiates or it helps out or it catalyzes the conversion of the various ribonucleoside monophosphates into ribonucleoside triphosphates. That is, adenosine monophosphate is converted into adenosine triphosphate. Accordingly, guanosine monophosphate is converted into guanosine triphosphate. So we can say that AMP gets converted into ATP. Clear? Then GMP gets converted into GTP. Then CMP, that is cytidine monophosphate, it is converted into cytidine triphosphate. And then last that we are talking about, that is uridine monophosphate, it gets converted into uridine triphosphate. Okay, so this is over here achieved with an enzyme which is known as phosphorylase. Clear? As soon as phosphorylation of these ribonucleosides occur, what happens that this DNA strand it unwinds. Basically, the unwinding of the DNA strand starts up with the attachment of the DNA dependent RNA polymerase enzyme, and as the RNA polymerase enzyme proceeds further on this particular DNA strand, what happens, the unwinding of the DNA molecule take place, thus exposing the hydrogen bonds between the nitrogenous bases breaks off, and it exposes the nitrogenous bases in the karyoplasm. Clear? So what happens, according to the phenomena of according to the principle of complementarity, what happens, these triphosphates, ribonucleoside triphosphates, they get attached to the the nitrogenous bases that have been exposed now in the karyoplasm. Clear? And that happens according to the rules that have been given by the Watson and Crick in their double helical structure. That is, A is going to bind up with U with a double bond. Clear? Then G is going to bind up with C with a triple bond. Clear? So all these ribonucleotides, according to the complementary base pairing, they get attached themselves to the various bases that have been exposed in the karyoplasm. These are the base pairs of the DNA template strand. And as the DNA processing, the RNA polymerase, it is proceeding further. What happens that using this RNA polymerase enzyme, these ribonucleotides, they get attached themselves to the preceding first nucleotide that has arrived earlier to them, and they form a long chain, polynucleotide chain. And now what happens that as soon as this terminator gene is reached up, this mRNA molecule that has been synthesized on this DNA complementary strand, what happens, this mRNA molecule, then it gets separated from the original DNA template strand. Clear? Now what happens, as soon as it gets separated from the DNA template strand, that is the mRNA molecule, the newly synthesized, the nascent mRNA molecule that has been formed, it gets detached from the DNA template strand and it gets floating away in the nucleoplasm or the karyoplasm. Now what happens, this DNA strand, it assures that it again winds up and it forms the base pairing with the original base pairing. That is, it is going to form base pairing with the base pairs of the antisense strands. That is, the hydrogen bonds, they are going to be regained back, and the DNA molecule, it is going to be regain its original form and structure, and the mRNA molecule along with the RNA polymerase enzyme, it gets separated from this DNA template strand. So here we can see that with the help of only a single cistone here, we can see that the mRNA molecule can be easily synthesized, and not only a single mRNA molecule, a number of the copies of the mRNA molecules can be synthesized from this particular type of a single cistron. Now the mRNA molecule that has been synthesized on this DNA template strand, it is not mature. It is going to contain both the exons and the introns, as well as I have said you all that the exons, they are the sense genes that are going to express themselves, that is, they are going to code up for certain type of the polypeptide chains. But the introns, as I said you all, that they are the non-coding gene sequences. So the maturation of the mRNA molecule has to take place, and the maturation of the mRNA molecule takes place by a couple of methods. That is, either it can be taken up by the capping method, that is cutting off the long polynucleotide chains, that is a long mRNA molecule into smaller forms, or it can be done by splicing. It can be done either by folding up of the mRNA molecule on itself, and it can be done by a couple of ways. Clear? So editing of the mRNA molecule takes place. In splicing what is done, that the introns, the intron genes, they are removed, and the exons, they are they are allowed to remain at the back, and then with the help of the DNA ligases enzyme, all these all the structural genes, they join up to form a complete mRNA molecule. So in this way, a complete mRNA molecule is transcribed on a single DNA template strand, or we can say that on a single cistrone of the DNA template strand. This is how mechanism of transcription takes place in the eukaryotes. Now, in my further videos, we are just going to talk about the process of translation as well as the genetic code. Till then, thank you.